everyone, and welcome back to the next webinar in our Plantay Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantay, the open online community for plant scientists, powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. We've held over 35 events in this series so far. If you haven't already done so, check out our archive to watch all of the previous talks in this series. You can find information about each event on our plantay.org blog and subscribe to our Plantay YouTube channel to get notifications when new recordings are posted. Next week, we will have our last Plantay Presents event in this series for the season. After that, we will be switching gears to plan and host the annual plant biology meeting online. If you need to leave this webinar early, know that a recording will be made available within a few days. Today's talks will each be 13 minutes, followed by a brief, short question and answer section. As you listen to each talk today, add questions for our panelists in the Zoom question and answer Q&A section. Questions specifically for our panelists should be shared within the Q&A section to make it easier for our moderator to navigate. You'll notice that there's also a chat area open. This section is for you to introduce yourselves, say where you are from, and connect with your colleagues. All right, so today is a special event that's featuring the work of 3 authors who have recently published in the Plant Cell Focus issue on plant genomes. Blake Myers, who is currently serving as a Plant Cell editor-in-chief, is with us today to share a little bit of information about the journal and this focus issue. Thanks, Blake. I'm going to turn it over to you now.
Thank you, Katie. So I'd like to welcome everyone to the Plantay Presents webinar, and thank you for attending today or for catching up on the recording later. This is the second of our series of talks highlighting papers from the focus issue on the biology of plant genomes. I'd first like to thank the editors who worked on this focus issue, including Nan Eckert, Jim Birchler, Siobhan Brady, Robin Buell, and Jim Liebensmack. As you may have seen, we have an outstanding lineup of authors presenting their work, selected in part by the editors from the larger group of papers. I'd encourage you to check out the full issue on the Plant Cell website. I'm really grateful that the authors have agreed to present their work, and I also encourage you to check out the last set of presenters in the recording from a few weeks ago. I am really pleased about the outcome of this focus issue, not just because it's the first focus issue for the Plant Cell, but because genomics is near and dear to my heart and my lab's work. It's really been a tremendous revolution in genomics tools and approaches, and there's a broad range of tools available now for modern genomics. This opens lots of interesting questions that investigators are asking, reflected in the outstanding group of papers that we've highlighted by these talks. We'll continue to add papers to the collection over the next few months that is on the website, compiling a resource for our readers. So please do check back to the website and keep an eye out for the e-table of contents. We do have other focus issues coming up. We're looking for outstanding work, so please send us your submissions. This includes a focus issue on cell biology with a submission deadline of May 1st, and a focus issue on plant biotic interactions with a submission deadline of September 1st. So now I'd like to hand it off to Judith Van Dingenen, our assistant features editor with the journal, who's introducing us today, and she's going to introduce the talks. So thank you all.
Thank you. Hello. So the first speaker of today is Jim Birchler. I hope I pronounced it correctly. He is a professor of biological science at the University of Missouri, Columbia, and he received a prestigious Curators Distinguished Professor Award in 2009. He also recently received the Barbara McClintock Prize for Plant Genetics and Genome Studies in 2020. And his research interests relies on structure and behavior of chromosomes, artificial chromosomes, centromere, epigenetics, heterosis, polyploidy, aneuploidy, using maize, but also Drosophila as a model organism. His lab focuses in resolving the mechanisms involved, the interconnection between genes and chromosome levels, and how they evolve. So today he will talk about how regulatory gene expression matrices affect expression, the phenotype, and evolutionary processes. The screen is yours, Professor Birchler.
Thank you very much for the invitation. It's my pleasure to talk about the topics of companion papers that will be in the special issue, focused on how regulatory gene stoichiometries affect plants. So the idea of gene balance is that partial genome dosage changes are generally more severe than whole genome changes. In humans, we are aware of this from Down syndrome, for example. And so this, what we learned in plants, applies across basically all eukaryotes. The idea of gene balance originated with Alfred Blakeslee, shown here in the Station of Experimental Evolution, which is now Cold Spring Harbor Laboratories. So he worked on the flowering plant Datura, and 100 years ago, he published this paper noting that each chromosome has an influence on the strength of expression of anthocyanin pigment. Some of them being lighter and some of them being darker, but if you have all of the chromosomes varied and have a triploid or a tetraploid, then the difference is not so great. So he actually coined the term of balance, and this has been borne out over and over again in many species, as we've noted. So in Datura, depicted here with the seed pod, this is the normal seed pod, and then if you add an extra chromosome to the genotype, it will have an impact upon the morphology of the plant, each one of them being characteristic. And so this is an illustration then of this genomic imbalance effect upon the morphology of plants. So some questions in gene balance could be, why does the dosage of every chromosome affect any specific morphological characteristic? Why does the dosage of every chromosome modulate expression quantitatively? Why is this not cumulative when the dosage of all chromosomes is changed? And why does the relative number of chromosomes determine the magnitude of the effect? So we work on maize, as my background might imply, and maize is very amenable to studies of genomic balance because you can make crosses to produce many different ploidies with extra or missing chromosomes. This plays off of the genetic tricks that are available in maize, primarily translocations between the supernumerary B chromosome and various chromosome arms. So this allows one to make diploids that have 1, 2, 3, or 4 copies of most chromosome arms. And then applying that with a haploid inducer, you can get haploids and haploids with extra chromosomes to investigate this in great detail. So here's a family portrait of the long arm of chromosome 1, where in diploids, if you have 1 copy of 1L, 2 copies in a normal diploid, or 3 copies, you can see that there's this impact in maize as well. These are haploids over on the left. This is a normal haploid, and this is a haploid that has an extra copy of 1L. And so you can see then that in the different ploidies, the relative number of chromosomes impacts the magnitude of the effect. So one can march around the various chromosome arms. Here is the short arm of chromosome 5 in the same kind of family portrait, haploid with 5S, and then 1, 2, and 3 doses in a diploid. And you can keep going with the shortest chromosome arm in maize, 9S, haploid with 9S, 1, 2, and 3 doses in an otherwise diploid. So you can see the general principles apply across the whole genome. There are differences, of course, as you might expect, which was also found in Datura. So what these papers that we're focusing on address is, what is the molecular basis of genomic balance? So many people have interpreted these types of effects as basically due to a dosage effect for the genes that are on the very chromosome arm. But as I will relate today, we believe this is centered around gene regulatory processes and has implications for the control of gene expression, quantitative trait genetics, and gene evolution. So what you'll see is that the major effect that occurs is what we call an inverse dosage effect, which we found in maize many years ago, looking at embryo proteins in a dosage series of 1L, 1, 2, and 3 doses here. And you can see some proteins are increased in 1 dose and reduced in 3 doses. We call this the inverse effect. This protein here is encoded in 1L, and it has about the same amount of gene expression in the dosage series. We call that dosage compensation. And there are some proteins, this one right here, that is affected positively by the dosage series of 1L. So we've studied this, as alluded to in the introduction, in Drosophila as well, and have defined the inverse effect and positive effects to the actions of single genes. And they are transcription factors, chromatin proteins, and members of signal transduction cascades. And what they have in common, I won't go into the details of it, but they have in common that they are involved in multi-component interactions for gene expression. So focusing now on the papers, what we did was conduct an Rna-Seq analysis, and this involved multiple biological replicates, and we cytologically verified each of them. Then we averaged the sequencing reads in the aneuploid, the individual chromosome variation versus the balanced respective ploidy. Separated the genes that were varied, we call those the cis genes, versus those in the remainder of the genome, which we call trans. Then we create a ratio of the aneuploid to the ploidy, or for different ploidies for each gene, and then plot those in a distribution, which is depicted here. So a ratio of 1 with the bold red line here would mean that there's no change. And so when you plot this here for 1 dose of 5S relative to the diploid, you can see some of them are reduced in expression, but some of them go up to the dosage compensation level. If you look in trans, there is a generalized trend for increased expression, this inverse dosage effect that I talked about. And so we then validated these with ddPCR and performed statistical analysis and differential gene expression. So you can do 1, 2, 3, 4 doses of 5S, and you can see when there is 1 dose, there is a trend toward increased expression in trans. When you have 3 copies of 5S, there's a trend toward reduced expression. And in 4 copies, you can see that there's a further reduction in expression across the genome. If you look at individual genes on 5S, you can find those that show a gene dosage effect. They're directly correlated with a copy number of 5S. There's another gene here that's dosage compensated, and you can find an individual gene here that is inversely affected in the 1 to 4 dosage series. If you compare ploidies, haploid, diploid, triploid, tetraploid, the ratio distributions in this case are much tighter. There is much fewer relative changes in gene expression, just as occurs with the morphological characteristics. But in a ploidy series, the volume of the cells are increasing through the ploidy series, suggesting then that the whole transcriptome size changes. And we performed an analysis to test the size of the whole transcriptome, and indeed, looking at the haploid, triploid, and tetraploid relative to the diploid, there is an increase in the size of the whole transcriptome, as has previously been found in other species. So in terms of the global studies in maize, as I mentioned, one can do a very great spectrum of aneuploidy to be examined in maize. Disomy versus haploid. In a diploid, you could look at monosomy, 1 dose, trisomy, 3 doses, tetrasomy, 4 doses for many chromosome arms. So here is just some cases of haploids and their haploid disomies for 3L, 4S, 4L, and 5L in the paper that is featured in this focus issue. Hua Yang was the first author, and the complete spectrum is shown in that paper. So what Hua was able to do was to analyze 17 regions of the genome of maize that comprise about 80% of the genome. And if we look at the effects in trans, which are the blue ratio distributions, you can see that for every one of them that have a downward pointing green arrow, which is all of them, there is a subset of genes that are in fact inversely regulated across the whole genome. There are a few with the upward pointing red arrows that have an increase also for a subset of genes that have a positive effect of that particular region. We analyzed the whole transcriptome size for this series, and indeed, for some of the regions, there is an inverse effect on the whole transcriptome size, those that are highlighted here. And in one case, for 6L, it even goes below the inverse level, which would be 50% for a disomic haploid. So now turning to diploids, if you add the chromosome arm to the genome, we could do that for 15 regions. And as you can see with all of the downward pointing green arrows, the effect across the whole genome is also largely an inverse effect. Some regions have a subset of genes that are upregulated, indicated by the red upward pointing arrows. And then by taking away a chromosome arm for monosomy, we could analyze 14 regions. And now we see with all of the upward pointing red arrows in trans, you can see that there's an inverse effect in those cases. But something new showed up here in the monosomies, and that is there was a substantial number of regions that have a positive effect in monosomy. And those are depicted by the downward pointing green arrows that have a positive effect in this particular dosage type. So how does this then affect gene expression? Well, we think what we refer to this as Kinetics Genetics. And so this plays off of regulatory interactions, usually are conducted by multi-component complexes. And so it plays off of a study by Bray and Lay a number of years ago looking about how the components of a complex will assemble. If you increase the concentration of a bridge subunit, you have an increase in the concentration of the whole complex that will be positively correlated. And then once you reach a peak, it will then at higher concentrations now have a negative correlation. So how do we imagine this on a very simplistic way? So if you imagine then you have a regulatory complex very simplistically of A, B, and C that's required for target gene expression, and you have those in a stoichiometric relationship that is very similar then as they assemble A with B and B with C before they form the complete complex, it can go to completion. But if you were to have a trisomy, for example, in which the bridge subunit is now increased in expression, you can see that it is not possible to go to completion for ABC, and the relationship then becomes an inverse relationship. So the overexpression of a bridge subunit then would have a dominant negative effect, depending on the order of assembly, which admittedly is not known for many complexes or interactors, which is a very important area for future investigation. But as we noted with monosomy, very oftentimes there was a positive effect. And so now if we can imagine the same scenario, if we now have a monosomy where a bridge subunit is now reduced to half of the normal level, now it becomes limiting on this side of the curve as opposed to on that side of the curve with an increased expression of the bridge subunit. So now underexpression of the bridge would be limiting, as is the case in some of the monosomics as we saw. So we can summarize all of this in this little expression here. Dosage determines whether you live or die, can't be too low and can't be too high. Changing the ploidy is usually not bad, but when it's aneuploid, it's really quite sad. Expression in ploidy, neither better or worse, but expression in aneuploidy is usually inverse. Some RNAs go up and some go down, but usually the latter is what we found. What is the basis? We think it's kinetics. We've addressed this problem by using genetics. So we visualize this. Our hypothesis to explain it all is what we call the gene balance hypothesis. This is basically stated by saying that varying the stoichiometry of multi-component interactions will affect the function of the whole as a result of the kinetics. Gene regulation and development, we know, is mediated by multi-component interactions, and therefore gene regulatory systems will be dosage dependent, and this will impact quantitative characteristics in a multigenic fashion. And we have also written that the differential retention of regulatory genes following whole genome duplication appears to mimic this aneuploidy-ploidy relationship that I told you about. And so in conclusion then, gene regulation will be influenced by balance principles. This suggests the need for many new studies involving this concept. The gene balance hypothesis, explaining this further, is in this publication here with Rainer Beedia, who is our thought partner in this endeavor. And so I want to acknowledge many contributors to this work as we conclude over the years and the funding from the National Science Foundation. Thank you very much for your attention.
Okay, thank you very much, Jim. I still need to unhide myself, but this is apparently not working. Ah, here. Okay, we have one question from Matthias Benoit, and he congratulates you with the great talk. You showed that the transcriptome size increases with ploidy, but did you observe an increase in transcriptome complexity too?
If you look at the individual genes that are expressed through the ploidy series, there is a very similar repertoire of genes that are expressed from haploid to diploid to triploid to tetraploid. In making the ratio distributions, you have to have expression in all of the different genotypes that you compare. And so this was something that we looked at. And so there was very few genes that were not able to be examined in a ratio distribution. So the repertoire of gene expression through the ploidy series is very similar.
All right. I think this was the only question that you got. Maybe there are more coming. Yes, there is another one. Does this mean that if you know the complex a protein is working in, you could predict the outcome or different ploidies for that gene?
One of the reactions that we often get to these results is that if one is doing an overexpression analysis, they often find results that they don't understand. And that probably traces back to this phenomenon of genomic balance. And so if you're looking at a regulatory complex and you do an overexpression analysis on your favorite gene, it could have a positive effect or it could have a negative effect depending upon where it acts in a particular complex. And so it's a tale of caution for trying to understand how the various components of a multi-component complex operate.
Okay, the questions are coming now. Wait, another question of Huawei Wu. I'm curious how in complex genetic network, how to locate them in the bridging points as in the multi-component interaction networks.
I'm not sure I got the question there.
He is curious how in a complex genetic network, how to locate them. Ah, yeah, okay, I understand. In the bridging points as in a multi-component interaction network.
So that's a really important question for future research. There's actually very little known about the assembly of complexes basically in any eukaryote. And so this, I think, the results of our study basically emphasize the importance of addressing those kinds of questions. So the order of assembly basically will have probably an impact upon what the target gene expression will be. And so it's a difficult thing to actually figure out is the order of assembly of a complex. But I think that is a really important future direction for studies of gene expression, considering the kinds of results that we talked about.
Okay, I think a last question that we can do from Mike Stam. Did you identify specific genes that drive the observed phenotypes when not in the proper balance?
So we have not done that in maize, but we have spent many years doing this in Drosophila, and we've identified individual genes. I alluded to it, and if you go back to those references that I noted, it will elaborate all of the individual genes that we identified. So in Drosophila, we can identify individual genes that produce both the positive and inverse effects that either reduce the expression of a target gene by 50% or increase it up to about twofold when mutant is a heterozygote. And in some cases, when we add the gene back to the genotype, it will have a negative impact upon the expression of the target genes. So those are, as we mentioned, those are typically transcription factors, members of signal transduction cascades or chromatin proteins. And those, at first, when we compiled all of those some years ago now, we were sort of curious about the heterogeneity of them all. But then if you read in the background papers that I noted, we note the evidence that those are members of multi-component interactions, and that seems to be the unifying theme to those genes, individual genes that produce the same kind of effects that that I told you about in our maize aneuploid series.
All right, I think we can end here. Thank you very much again for the talk. And we can go to the next speaker. So the second speaker of today is Evan Forsythe. He is a postdoc at the Colorado State University in Dan Sloan's lab, studying evolutionary processes at the molecular level. Since 2018, he's a postdoc there, and before he did his PhD with Mark Beilstein at the University of Arizona, where they focus on comparative evolutionary biology. And his research even uses phylogenomics as a tool to understand cell and molecular biology, including the functional impacts of hybridization, duplication, and plastid nuclear co-evolution. So today he will present his recent published work, where he also uses phylogenomics to detect genome-wide signatures of plastid nuclear co-evolution point and genetic interaction. Okay, the screen is yours, Evan.
All right, thank you. Yeah, let me know if you can't hear me. But so today I'm going to talk about plastid nuclear coevolution and a method called ERC that can be used to identify functional interactions between genes. Sorry, I'm just getting my pointer out because I think I'll need it later. So plant cells contain their DNA in 3 different compartments: the nucleus, the mitochondrion, and the plastid, or the chloroplast. So each of these has the ability to encode its own proteins. And the function of the plastid, for example, relies not only on the proteins encoded in its own genome, but also the proteins that are encoded in the nucleus and then imported into the plastid. So most plastid functions rely on plastid nuclear complexes, like the one shown here. This is the caseinolytic protease, or the CLIP complex. And so this is just really one example of the remarkable level of coordination that's required between the plastid genome and the nuclear genome. And this level of coordination is remarkable because these genomes are quite different. They undergo different levels of mutation. They experience different levels of selection, and they ultimately evolve very differently from each other. For example, if we look at a tree, this is a tree from 20 angiosperm species showing CLIP-P1, this plastid subunit. And the branch lengths here are showing amino acid substitutions. And what you can see is that this complex is really highly conserved in most species that we're looking at here, but in a few independent cases, in a few independent lineages, you see this really dramatic acceleration in the rate of protein evolution. So this protein has changed just a ton in some of these lineages. And this phenomenon is not specific to CLIP-P1. Shown here are 6 additional plastid genes, or groups of plastid genes. And these trees also exhibit evidence of accelerated evolution in some lineages. So this is just a really dramatic example of accelerated evolution. And it's really a mystery. What are the evolutionary mechanisms that underlie this type of evolution? And it's especially confounding given that each of these proteins at some level is interacting with genes encoded in the nuclear genome. So let's just consider the evolution of an entire complex by just following the evolution of this cartoon CLIP complex over these hypothetical species. You can see that as this complex evolves over time, if you have one of these periods of accelerated evolution in CLIP-P1, you might also expect selection to favor compensatory mutations in the other subunits of the complex in order to maintain their cooperation. And so if we were to infer a gene tree for each of these types of genes, we would expect to see this mirroring effect in which we have a long branch in species Y in both trees. So this evolutionary pattern is called evolutionary rate covariation, or ERC. And this has indeed been demonstrated between the different subunits of CLIP and also among other groups of known interacting proteins in plants. And this concept has been extended in model systems outside of plants to the whole genome scale, meaning they can apply this type of analysis to scan the entire genome to identify interacting genes. And they've used this type of analysis to identify interactions within complexes, but also importantly, this technique has been used to show that co-functional proteins, these are genes that contribute to some shared process without necessarily physically interacting, can also exhibit ERC. So This makes ERC a really powerful tool because it has the potential to identify genetic interactions at multiple different levels. And the tool is also quite scalable, meaning there's at least the potential to apply ERC widely to identify these large interactome networks. But this tool has not been applied at the genome-wide scale in plants previously. And the main reason for this is that plants undergo a lot of gene and genome duplication. And this creates challenges for phylogenomic studies because most gene trees have gene duplication events in them and paralogous sequences. So we set out to overcome this challenge by developing a pipeline that really briefly applies this technique called gene tree species tree reconciliation in order to identify the duplication events within trees. And our pipeline sought to remove the ancient paralogs that can really be the most challenging and problematic for this type of analysis. But importantly, we want this pipeline to be able to retain the more recent duplications because these are less problematic in the context of ERC. So we applied this pipeline in those same 20 angiosperm species I mentioned, and we were able to identify about 8,000 gene trees that are suitable for performing ERC analyses. And so with this technique, we're finally able to perform genome-wide ERC analyses in which we can make a comparison of a plastid gene against all of the nuclear genes in order to identify those that show evidence of coevolution. And I will say that we performed this analysis using 7 different plastid genes, but to present all those data becomes a little bit cumbersome in a short talk like this. So I'm going to continue to focus on the results from CLIP-P1, but I will say there's a decent amount of overlap between the results from different plastid genes. So I'll frame our kind of takeaways in the context of general plastid nuclear coevolution that is not necessarily specific to any one plastid gene. And so when we applied this, the first thing we did is we pulled out CLIP-P1 and compared it to its one of its known interactors, CLIP-R1. So this is one of the nuclear genes that's known to be in the CLIP complex. And as you could see, we see this really clear mirroring effect that is expected among interacting genes. But I'll also point out that CLIP-R1 in our data set has some duplication and missing data. So this is just goes to show that it is important that our pipeline be at least a little tolerant to some of this type of messiness in the data. Otherwise, we wouldn't have any genes to study. So when we can quantify this signature of coevolution by doing a correlation analysis. So in this plot, we're showing the branch lengths from CLIP-P1 along the x-axis and the branch lengths from CLIP-R1 along the y-axis. And as you can see, there's this really clear positive correlation. And we can quantify this correlation, of course, using p-values and r-squared values. So this is telling us that we are seeing this is telling us that we are seeing this ERC signature among known interactors. So we apply this to the entire genome, and here's what we see. So I'm showing on this plot, I'm showing both the p value, and it's a negative log transformed, and also the r-squared value. And on this plot, that means that the strongest signatures of ERC, those genes exhibiting the strongest signatures, will be in this upper right portion of the graph. And so this plot should have 8,000 points on it because we're looking at ERC in 8,000 nuclear genes. And I've also showed I've highlighted the known CLIP subunits, and I've even broken it down by their specific location within the structure of CLIP. And so what jumps out right away is that it does appear that the known CLIP subunits are highly enriched towards the top of this graph among the top ERC hits. So this is telling us again that ERC, even on a genome-wide scale, is returning the known interactors. But the next question is, what are all of these other gray points that are not known CLIP subunits yet exhibit really strong ERC signature? So we sought to learn what these subunit or what these hits are. And the first thing we did was we did an enrichment analysis using these top hits. And what we saw is that these top ERC hits are indeed enriched for being plastid localized. We also found that they showed an even stronger signature of enrichment for being plastid localized and involved in plastid nuclear complexes. And then finally, we saw depletion in non-organellar localized genes. So this goes to tell us that what ERC is returning is a real meaningful functional signal, and that's even against a genome-wide backdrop. And this is exciting because this means that the composition of the ERC hits that we're seeing has the potential to tell us about what specifically is driving plastid nuclear coevolution, and maybe even tell us a little bit about the about what is underlying those mysterious accelerations that I described. So we delved further into this. Whoops. We delved further into this by performing gene ontology enrichment analyses. And it just so happens that we saw that certain pathways or processes were especially prevalent in our enrichment analyses. And those were, we saw that plastid translation was especially prevalent among enriched ERC hits, and also import of proteins into the plastid, and protein quality control and turnover in the plastid. So all three of these processes were especially prevalent among enriched Go categories. And this is interesting because all three of these processes contribute to a higher level process, and that is the maintenance of plastid proteostasis. So this is maintaining proper protein levels in the plastids. And so it really seems, and this is an important process as evidenced by a mutant in plastid proteostasis. This is another gene involved in degrading old proteins, shows this really well-defined variegation phenotype. And so it really seems that plastid proteostasis is somehow driving plastid nuclear coevolution. And the most striking example of this comes from the fact that we also observed enrichment of cytosolic ribosome subunits among top ERC hits. So it seems that the cytosolic ribosome subunits are also co-evolving with the plastid. And this is surprising, of course, because the cytosolic ribosome is not located in the plastid. So how is it contributing to a plastid function? Well, it also makes sense if you think about it, because most of the proteins that end up in the plastid are synthesized out in the cytosol. And so it absolutely is necessary for the two translation systems to be coordinated in order to achieve plastid nuclear stoichiometry. And so this was an especially exciting result for us. And it was also very satisfying for us to go into the literature and find genetic or functional evidence of this phenomenon, in that a cytosolic ribosome subunit mutant serves as an enhancer of this same variegation phenotype that I described earlier. So the takeaway from this is that it appears that large suites of proteostasis genes all underwent accelerated evolution at the same time in some angiosperm lineages. So we propose that this means that proteostasis systems have somehow been perturbed in these lineages. And this perturbation somehow caused shifts in selection that simultaneously affected numerous functionally related genes. And so in our discussion of our paper, we discussed some of the possible mechanisms that underlie this perturbation. For example, rewiring of proteostasis systems, stoichiometric pressure caused by genome duplication, genomic instability of plastomes, changes in abiotic or biotic stress, and antagonistic interactions between the nucleus and selfish genetic elements in the plastid. And so please feel free to read our discussion of each of these possibilities and help us solve this mystery of what is underlying this accelerated evolution in plastid proteostasis systems. So with that, I'd just like to thank the entire Sloan Lab, especially Alyssa and Dan, who co-authored this paper. I'd like to thank the organizers and the editorial board for making all of this possible. And finally, our funding. So with that, I can take any questions that you have. Thank you.
Thank you, Evan. For the moment, there are no open questions. So everybody that has a question, you can just pop it there in the Q&A session. I have a question. I'm not so familiar with all these things that you are now presenting, but what I was wondering is, do you see that any of the plants that you study, where you study this plant nuclear coevolution point, is different and is acting differently than other ones?
So yeah, like are there specific things about the organismal biology of these plants that might underlie this? So the sampling in this study, we used all species that have whole genome sequences published, and it's widely sampled across all angiosperms, and so of course there's many biological differences between the plants that we're using. They're quite, they're meant to represent diversity as evenly as possible. We have not necessarily like pinned any, made any connection between some specific trait that's changed in any of these species and the evolution that we're looking at. But that would be an area of future work. That is where we want to move to. Like, what is it about these plants that has changed that has resulted in these inferred perturbation events that we propose?
Okay. There's one question. Lydia Gramzo, can you use this method to detect interaction between nuclear genes?
That's a great question, and yes, you can. And we have some ongoing work where we are doing just that, using this kind of as a proof of principle that yes, it does seem to return functional interactions. So we would like to expand this to the entire nuclear genome, and it's ongoing work, but I'll put out a plug for keeping an eye out for where we're planning to create a database that any molecular biologist could use to search their favorite gene against and return all of its interactors.
All right. Another question from Dong Wang. On the opposite spectrum, how about interspecies coevolution?
Yeah, so that I think that there are a lot of parallels between coevolution between genes and interspecies. For example, the accelerated evolution that is seen in the plastid has been proposed that it mirrors like host parasite dynamics or some sort of like arms race dynamics, because that's the level of acceleration that we're seeing here. Those types of mechanisms are what can drive that level of interaction. So that's just one example of how this mirrors interspecies coevolution.
All right. The questions. Okay, a last question maybe. From Sophia Zabal. Do you find that genes sometimes don't have a strong signal in ERC, or do you always find significant hits?
That's a great question. There's plenty of examples of when there's not a strong signal, even among known interactors. So this is not a surefire way of detecting things. Sometimes the analysis is just underpowered for a variety of different reasons. The most obvious reason is that we are studying as many genes as possible, but we are still filtering out some of the genome because it's just not suitable. These genes are not suitable for ERC analysis, meaning they maybe have undergone some wonky evolution that just make them difficult to study. So for those genes, we can't even start to look at the coevolution of those genes. So that's an example of some of the limitations of this.
All right, there's still one question, so maybe you can answer it during the chat and we go to the other talk because it's already 10 before 5, I think.
Sure, thanks for all the questions.
Yeah, thank you. Okay, so the last speaker of today is Tom Jacobs. He is an expert scientist and group leader here in Belgium in the VIB UGent Center of Plant Systems Biology. His group is focusing on the development of new genome editing tools for plant biotechnology in plant crops and model species using mainly the CRISPR-Cas editing tool. He did his PhD in the University of Georgia in the lab of Wayne Parrott, and he did his postdoc with Greg Martin at the Boyce Thompson Institute. Today, he will talk about CRISPR screens in plants, approaches, guidelines, and future perspectives. Okay, Tom, the screen is yours.
Okay, thank you, Judith. Can you hear me all right? Yep, okay. Yeah, thank you for the kind introduction, and also like to thank ASPB and Plante for giving me the opportunity to share with you our recent review article in Plant Cell. So as Judith mentioned, my lab, we develop genome editing tools and technologies specifically for plants. And one area that we specialize in is the development of CRISPR screens. And so last year when the lockdown started, we decided then was a good opportunity to dive into the literature and really dissect the reports of CRISPR screens in plants. There had been a few that had come out by that point, and we really wanted to kind of get a snapshot of where the field stood. Both for our own research, but we also thought it would be useful for the larger plant science community. So this was written with Christoph and Ward, two postdoc and student in my group, who are developing two different types of CRISPR screens in the lab. And so I think this technology is really interesting and powerful, and will be widely used in the years to come. So I hope that many of you find this seminar and this review article very useful for your own work. Yeah. So first, just to bring everyone up to speed on what I mean by a CRISPR screen, I think everyone knows what a standard CRISPR-Cas experiment looks like. We have a Cas9 protein paired with a guide RNA that directs the nuclease Cas9 to a target location to induce an insertion or deletion at a target sequence. So in our typical CRISPR-Cas experiments, we would deliver this genome editing reagent into a plant transformation pipeline and recover a number of independent events, all with the same or similar types of mutations at that target site or target sites. So by comparison, a CRISPR screen is really genome editing at scale, where we're delivering large numbers of different guide RNAs to target different genes within the genome. And so by delivering this large assortment, we, in our transformation pipeline, independent events that have one or a few different tDNAs that have been integrated and are knocked out for different target genes in different combinations. And so what this allows us to do is create a mutant population from a single transformation or a limited number of transformations and capture really a full mutation spectrum on some number of target genes. So CRISPR screens have been largely developed in mammalian cell systems and have really taken off in that field. The first report was in 2014, really standing on the shoulders of what had been done with small RNA library screens in the years before. So I thought this was a really fantastic paper at the time. It demonstrated designing guide RNAs genome-wide against all genes in the human genome, high throughput guide RNA construction on oligo arrays, and then transfection or transformation into millions, if not billions, of mammalian cells that resulting in a mutagenized population of cells that are essentially identical except for the guide RNAs that they carry and the mutations that have been induced by those guide RNAs. What is really useful with the CRISPR screen is that the guide RNAs are different in each target site, and these can serve as barcodes, as molecular barcodes, to track the abundance of the different cells in the screen. So after treatment with a chemical or a drug, researchers can screen for enhanced resistance or susceptibility within the population. So I thought this was really cool technology, and we could apply many of the same features for plants. For example, we have large numbers of gene lists that we would like to knock out or perturb in various ways. Typically, we are utilizing just a small number of target genes from our list, but using a CRISPR screen, we could very easily design guide RNAs to the entire sequence, order up oligos for all those guide RNAs, clone those into agrobacterium, and then transform those en masse into plants. So again, resulting in a mutagenized population of primary transgenics that could then, for instance, be screened under different conditions in certain cases like pathogen resistance or susceptibility. And because the guide RNA serves as a barcode, we could sequence the guide RNAs from each of these plants and then discover the underlying genes that are causing either susceptibility or resistance. So this is a table from our review. There have been now a number of CRISPR screens that have been performed in plants, CRISPR knockout screens for the most part. And there's even one late edition here that didn't make it before our acceptance being reported in Cotton. What I hope you can see here, there's really still just a handful of knockout screens that have been done in plants, and they really range in their size. So either from the tens of targets all the way up to a genome-wide knockout screen that had been done in rice. And so far, we've really just been using one or a handful of guide RNAs at a time to make the mutations. And so each of these reports, we've been able to identify mutant phenotypes in the resulting populations and associate those with the underlying genetic causes quite rapidly because we're just using that guide RNA barcode. There are other types of CRISPR screens that have been reported. For instance, base editing can be used for a sort of in planta directed evolution. So base editing relies on a DNA deaminase that is fused to Cas9. And rather than targeting a panel of genes, you can target the entire, do a tiling targeting across an entire single gene. So in this case, this is the ALS1 gene in rice, and all possible guide RNAs were designed. ALS1 is a very good target because there are known point mutations that confer resistance to herbicides. And so after tiling all the guide RNAs and all possible base substitutions, the authors were able to screen on a herbicide-containing medium and select resistant events. And from this approach, they were able to identify 6 variants that gave resistance to the herbicide, and also discovered one new variant to rice. In addition, we can also perform promoter bashing experiments that relate to engineer quantitative traits. I think the lab of Zach Littman has really developed this technology quite well by targeting promoter regions of their genes of interest. So designing a number of guide RNAs that along the span of, say, a 2 kb region upstream. And so by this type of targeting, different types of deletion mutations can be observed between the guide RNAs or even local mutations at guide RNA target sites. And what this results in is a spectrum of mutation profiles and expression from this target gene that results in really being able to fine-tune quantitative variation in tomato. So the second part of our review covers more of the how-to and practicalities of making a CRISPR library. It is a fairly new technology. Many things are still in development, and people are looking for ways to implement this in their own lab. So we present the general workflow and some design parameters that people should keep in mind, as well as quality control metrics that should be used throughout the entire experiment. So in the first half of the experiment is the design and creation of the guide RNA library. So there's target design, cloning, and then transformation of the material, either stable plants or cell lines. And there are a number of decisions that have to be made along the way, in particular number of genes or guide RNAs that are going to be used. One must also be sure to validate the genotyping assay ahead of time. I can't stress this enough. There's really no point in making a mutation if you cannot genotype it. Other key factors are making sure we have uniform coverage and uniformity of the guide RNAs within the library, just to make it a more efficient process. And of course, other standard systems like how we will deliver the reagents and calculate efficiency. Then once the population has been made, again, we also need to evaluate for coverage and uniformity to make sure that we have a well-represented library. We're not just screening the same 3 genes over and over. We also need to figure out how to screen the line, either the number of traits we'll go for, or how we'll do the phenotyping. Perhaps a reporter line would make more sense in certain situations. We also then need to figure out how to do the genotyping and phenotyping, and then connecting those two together to reveal the association between the phenotypes and the genotypes. So these are the general process, and one has to consider each of these steps before even beginning to design the guide RNA library. One practical way that we try to organize the constraints of our targeting experiments is just through a simple working equation that we use. So often when working with other groups, we are trying to figure out how many targets we can go after at once. Ideally, everyone would go for genome-wide, but that's really not practical in a lot of situations. So we have to consider a number of different features. So for one, is population size. You really have to be honest with yourself about how many you can possibly screen at a time. This is an example from my postdoctoral days, where just a screen on 20 different lines easily filled a greenhouse. So in something like tomato, it'd be very hard to do very large screens. Even in Arabidopsis, we face the constraints of greenhouse space for some of our approaches. One must also consider the mutational efficiency of the particular CRISPR system that's in use, either for knockout or base editing or any of the other approaches that I mentioned. So for example, this is something that we recently published in PBJ, showing different indel efficiencies at T1 in Arabidopsis. And what you can largely appreciate is that about half of our lines are highly mutated, the other half are not. And so this means we have to double the size of our transformation population if we want to target everything that we are hoping to get. We must also keep in mind that we're doing a random sampling distribution with our different T0 events. And so we need to make sure that to cover all of the different lines, or all the different gene targets, we have sufficient numbers of events that we're capturing. So typically, we sample about 3 times as much as we need to get about 90% coverage of our gene targets. And then the last factor is having sufficient replicates. So it's important to have independent lines with same mutations or combinations of mutations within them. In my own experience in tomato, I got very excited about some single line phenotypes that ended up just being somewhat clonal. So it's really important to make sure that the screens have replicates. If you find one really interesting line, it's risky to believe that one event. Another limitation when designing CRISPR screens is that it's not always possible to just use the guide RNA as a proxy for the mutation. So in the beginning, I told you that the guide RNA is a barcode. And indeed, this is typically how it's used. But during my work in tomato, I found that was not always the case. So what I'm showing here is just a small excerpt of one of the tables for a few different lines at different target genes. And so what I found is that some lines contained mutations. These are the little red boxes. Some genes contained a guide RNA, but they were not mutated. Those are the gray boxes. Then what was really surprising and interesting was that some lines contained a mutation at a target site, but this guide RNA was not present in the plant. And so the only way that this could have happened is if a tDNA got into the plant, expressed the guide RNA, induced the mutation, and then was degraded without integration. So I call these transiently induced mutations. In total, about 20% of the mutations that were found in tomato were caused by these transient effects. But this is not alone to just my report. It's also been observed with base editing with Kwong et al. And also other reports in the field using RNA delivery or protein delivery all make use of transient expression of reagents. So this uncoupling of the guide RNA from the target site is actually very problematic for plants compared to other systems because it means that we have to genotype a lot more than just the guide RNA. It makes that a more costly and expensive endeavor. Right now in my lab, when we do screens of about 100 genes, it takes us a couple of months to design all the amplicons to properly genotype all the targets we want to go for. So it's a very large upfront investment cost doing that. And I also expect that many of the CRISPR libraries that have been reported are actually much more mutagenized than are currently appreciated because many people are just sequencing the guide RNAs and not looking at all possible target sites from the pool. So as I mentioned, this is a severe limitation, having these transient effects and then the genotyping burden that we have. And so in my lab, my PhD student has been developing a pipeline, an automatic pipeline to help with the design analysis using available programs like CRISPR and Primer3 to find guides and amplicons and then find the ones that overlap in the sweet spot that we're looking for. So we've now tested this on a number of genomes of different species and have found a pretty good success rate, except of course in cases where there are a lot of duplicated genes, it's not possible to design specific guides or amplicons, but we can get pretty good coverage of our target genes in a pretty small amount of time. As I mentioned before, it used to take us a couple of months to design targets for 100 genes. We're now down to 40 minutes. So I think this will be really good going forward. We also need some new tools to improve our guide RNA prediction efficiency. So there are a lot of tools that are out there to predict on-target guide RNA activity. But unfortunately, at least in our hands and others in the literature, they've been really unreliable in plants. We don't see any clear correlation between activity in predicted activity and implanted activity. So we think using some of the pooled strategies that have been used elsewhere in the literature could be used in plants to uncover guide RNA efficiencies. Also would be interesting to use arrayed strategies. So everything that I've talked about today has used a pooled transformation approach, but we could also think of doing arrayed CRISPR screens in plants, for instance, using protoplasts and 96-well or other multi-well plates, followed by some sort of simple readout. Then just to end with some different future applications where we see this technology going, I think one really cool application will be with single cell screens combined with CRISPR screens. This has already been done in the mammalian field using techniques such as drop-seq. And so we can really start to dissect different cell types with the mutagenesis power of CRISPR screens. One limitation, though, right now with this approach is that at least with protoplasts, we need to develop a technology where only a handful of plasmids go in at a time, because currently with our current PEG-mediated transfections, they essentially all go in together. And then what I think would also be interesting in relation to the previous seminar that we saw will be genetic interaction screens. So this is an area that I'm very interested in developing in my group. So far, as I mentioned, we've just done one or a handful of targets at a time, but we can easily multiplex 10, 12 guides at once and make very large combinatorial collections of mutants. The combinatorial space is huge when we do this, so we'll need to develop new modeling methods to be able to utilize what we can screen and learn more about genetic interactions in plants. So with that, I just need to close and thank everyone that's been collaborating with me over the years, helping develop CRISPR screens. As I mentioned, there are a number of ongoing projects, so please stay tuned. There'll be a lot more to come. And I'll be happy to take any questions.
Okay, thank you, Tom. There's one question from Farhan Ferozi. Have you tried CRISPR knockout screening on Physicometrella patents, since it doesn't fill the greenhouse and it is a good model system as far as genetic redundancy is concerned due to its dominant haploid stage?
That is a very good idea. I recently submitted such a proposal, which is why I'm laughing. So yes, I am considering that. There, I think the difficulty with the PEG-mediated transfection would come into play. It would be harder to do a pooled screen because you just have so many millions of copies of plasmids per cell. So an idea has to be found around that.
Yes. There are no other questions for the moment. So everybody that still has a question, you can put it in the Q&A box. There's still some time. There is one from Mukesh Lotha. Is it possible to do screen and Arabidopsis with floral dip methods?
Yes, so we're doing that right now. Yeah, it depends on the scale that you want to go. I really think where CRISPR screens shine is with the multiplex knockout. At least with Arabidopsis, there are plenty of single gene knockout resources that are available. But if you want to overcome genetic redundancy or make higher order mutants, I think that's really the application in Arabidopsis.
Yes. Farhan also wants to do a PhD in your group. He just mentioned in the chat. So if you have an open position, you can contact him.
I think the position's still open if you want to if you want to apply.
Perfect. No other questions for the moment. So that's it, I think. Yeah, I think that's it. Thank you, Tom, for the presentation.
Yeah, thank you, Judith, and thank you, Katie. Katie, you seem muted.
Whoops, sorry about that. I was saying thank you all for some great talks, and also Judith for the excellent moderation. Thank you all for attending, and we will see you at the next Plantae Presents event. Bye, everyone.